Hi everyone, thanks for tuning in. My name is Matea Djokic and I would like to talk about a fat measuring device called the Fatmeter for use in measuring fat of a species of fish called pallid sturgeon. Pallid sturgeon are affectionately known as a dinosaur fish because of their dinosaur-like appearance and the fact that they've been around for 70 million years. They have smooth skin rather than traditional fish scales, and they have bony protrusions along the sides and tops of their bodies. They can live up to 100 years and they can grow up to 6 feet long, which is huge. Pallid sturgeon are native to the Missouri and Mississippi rivers, and they were declared endangered in 1990 due to several factors, including overfishing and dam construction. They are also no longer able to reproduce in the wild. Because of this, managers and biologists began programs to artificially raise fish in hatcheries and release them into the river. These programs started in 1998 to stop the species from going extinct. The fish from these programs are surviving after they're released, which is great. But recently, we've seen that the growth of these fish has slowed compared to growth of fish that were released in earlier years. This graph is showing that fish released in recent years are growing slower than fish released in earlier years. Age is on the x-axis and length is on the y, and each of these lines is representing a group of fish that was released into the river in a given year. So here you see that most of these fish are growing at similar rates until they hit about age 6, where fish that were released in 2002 start to grow faster and larger than fish released between 2006 and 2009. There are several hypotheses or explanations as to why we're seeing these huge differences. One is that we overstocked the ecosystem, meaning that we just put too many fish into the river and they are competing for the same food. This means that the fish cannot eat as much or store as much fat to grow at the same rate as fish that were released in 2002. But managers don't have the tools to understand what is causing this change in growth or what it means for fish health. So you might be asking, if that's the case, how can we possibly measure the health of these fish? One tool used in other species is called the Fatmeter. Pictured here is the Fatmeter, and when it's pressed against the skin of a fish, it can estimate its fat content. It works well in many species of fish, including salmon and catfish, but it hasn't been used in pallid sturgeon before. This is where my master's research comes in. I looked at whether or not the Fatmeter would work for pallid sturgeon. And here's a graph from my master's thesis that shows the relationship between Fatmeter measurements and percent fat. Fatmeter measurements are on the x-axis and percent fat is on the y. And there is a relationship between these measurements, but Fatmeter measurements alone only explain about 3% of the variation in percent fat of the fish that I measured in my experiment, which means it didn't work very well on pallid sturgeon. And there are a few reasons why I think the Fatmeter didn't work as well on my fish as it did for other species of fish. First, those bony structures that I spoke about earlier may interfere with fat meter readings. Also, the fat meter may only capture muscular fat, which is an issue for pallid sturgeon because they store most of their fat in their internal organs. Lastly, the fish in my experiment may have been too small. I measured fish that were much smaller than the ones in my photos. The ones in my photos are adults, but the ones in my study were juveniles or teenage fish. I saw that the fat meter generally worked better on some of the larger fish in my study. This study was important because it showed that although the fat meter worked well on other species of fish, it did not work well on the size of pallid sturgeon in my experiment, and that more research should be done on larger fish to see if this fat meter could be more useful for bigger pallid sturgeon. I'm so glad I could talk to you about my favorite fish today. Thanks for joining me.